Only mode.
Good morning, everybody. Welcome. We're pleased to have you here for what I think is going to be the last in this series of webinars for the RA community phase. Today, we really just want to go through talking to each of the groups and the teams specifically in the groups and hear what your experiences have been, what's working for you, what's not working for you, what you might be having difficulty with, and see if we can support each other, because as you can see from my screen here on the leaderboard, we still have not succeeded for challenge question one in coming together in the community phase to boost the score above what was seen by the Guan Lab in the team phase. If we look at the leaderboard for question two, we are seeing a couple of submissions which are better than were in the team phase. So that's a start. But you know, one of the rationale for the community phase was you're all intelligent people with overlapping expertise, and perhaps by coming together and talking, you will come up with something where the sum is more than the individual parts. And so we'll see if we can facilitate that a little bit today. And really, I'd love your thoughts on whether you think, or how you think this is going, and what you think might be the outcome at this point. We're planning to run the community phase through mid-September, which means I will send out detailed instructions this week on how we're going to end this. But the idea was that we would have a final scoreboard submission twice. Once in the beginning of September, that would be a direct comparison against the scores that you all had in the team phase. And then we're going to release the data from the leaderboard so that you have a slightly bigger sample size to work with, and then ask you to submit to the final scoreboard a second time 2 weeks later, so we can see if that helps boost power at all. Okay, with that, I'm really looking to all of you to lead discussions today. So I'm going to go through the groups and ask you to answer the 4 questions I sent you in the email, which is, those questions are, what have your experiences been in selecting features based on biological priors? Have you considered approaches for polygenic modeling? What are your thoughts on ensemble methods? And has working in a group helped you? Has the collaborative approach had any effect or influence on your modeling efforts? So we're going to go down the list. We're going to start with group G. Is it GTSM? And so Lou, I'm going to put you on the spot. Then Tao, Wanfeng, and Javier. So let me find, Lou did have some slides, so let me find those. Not those here. And so I'm going to unmute you, Lou. There you are, we can hear you now.
Okay.
So how's it been going for you?
Okay, cool. Yeah, it's going on, but then we didn't really make too much progress. It kind of we are trying lots of experiments, but then now the experiments are really promising yet.
Yeah.
Okay. So could you open the slides?
Oh yeah, sure. Can you see them?
Yeah, I can see them.
Okay. Oh, my screen just went black. It's not good. There it is. Okay.
So could you go to the next slide?
I am trying. What is going on? Huh.
Okay, so the first thing we did is like we repeated the experiment using gamma with other team SNP sets. So this will tell us whether this is because of the method or because of the SNP set which performs better. And so previously we were just predicting for the whole. Hello, could you hear me?
Yes.
Okay. So there seemed to be some connection problem on my screen. Okay. So we previously we were predicting for the whole patient, but now we made some modifications. The first is like we stratified according to the drugs, stratified the patient to the drugs, and then we train a model for each different groups which are by the drugs, and then the gamma results are really similar to the original teams. So for the GuanLab SNP, we got 0.56, and then on subchannel 2 it's 0.74. And then GuanLab got like a 0.52, something like that. So the results we think like are really similar. And then also the same thing holds for SBI Lab and then Tracy team. So, but for Tracy there is something special because they didn't use drug specific specific SNPs for each groups. So we just use a whole, we just stratify the patient and using the same SNP set for all three groups. Next slide, please. So the previous slide shows that the SNP sets are really valid because we can predict using different methods, and they are providing similar results. And then next validation we want to take is like the covariate. We predict only using the covariates. So actually when we are doing the stratification, the performance increases for sub-challenge 1. So previously the performance was 0.45 in using only the covariates, and then it goes up by 1%. And then for sub-2 it's like quite similar. It's a little bit less, but then I think they're really similar. So this means the stratification also helps here. What we did is we also selected random SNP sets together with a covariate to make the predictions, and we found that the random SNP set deteriorates the predictions. So basically, what we do is like we select 50 SNPs from the whole set of SNPs, and then we combine them with a covariate and feed them into gamma, and then we predict. And then the experiment was repeated 10 times. All the predictions are worse than the covariate only prediction. So the average was 0.48, and then the covariate only prediction was 0.427 in the cross validation. We didn't submit to the leaderboard because we want to save some submissions. So we will expect the same similar kind of behavior if we submit the result to the leaderboard. Next slide, please. So meanwhile, we also tried other experiments. So one idea is like we test a single SNP versus multiple phenotypes. That is the baseline and the delta DS, and then the binary phenotype. So this is based on canonic correlation analysis. The idea of that, and then we selected 29 SNPs for the prediction, and then the predictions are really similar to the covariate only prediction. I don't know whether this is a good SNP set or not, because if we choose a random, it's much less. But then for this SNP set, it doesn't really deteriorate too much. But it doesn't really also improve the performance. So maybe there is some information, but then currently we can't really improve on the current results. And also we extracted the drug-related SNPs from the PharmGKB database for the prediction, and then the results is the same. So it's also slightly worse than the covariate-only predictions. Yes, next slide, please.
Lou, can I ask you a question about this first analysis, just to make sure I'm understanding? You were looking at associations between individual SNPs and you said baseline DAS and delta DAS. So you're looking for
Yeah,
SNPs that were associated with both of them?
yeah.
Hmm. Um,
And also the phenotype, uh, the binary phenotype, the responder and non-responder.
yeah. Yeah, okay. So, and it didn't do any better than covariates only, is that right?
Yes, it don't. It's just slightly worse, actually, slightly worse.
Yeah. I guess I would imagine that a SNP that has a strong effect on baseline would be negated by the clinical, would be represented by baseline DAS in the clinical model um, itself. So
Yeah,
that,
I mean, this is like a hard to say. We just need to try lots of experiment, and then sometimes some teams perform slightly better, and some don't perform really good. So I hope that in the following experiment, when we combine them with a good percent, and they can perform better. But then I think other teams like SPI, they say that they combine their SNP set with a GoLab SNP set, and then the performance doesn't really improve. So so we don't know. We just need to try.
yeah. Okay. Uh, yeah.
So
Oh, here's my
so
four questions. Great.
yeah. So the discussions about using biological prior for the feature selection, yes, we did, and unfortunately, uh, don't really improve the current predictions. And then a polygenic modeling approach. Actually, the gamma is one of the references that you have pointed out, and then we have been using all the time, and then we are making the stratification, and then there are other things, and also doing the cross validation. So we have been, I think, the method is performing well, and but just I think we need to focus more on the SNP sets. Use of ensemble method. Actually, we have proposed that we will use ensemble method, but then the initial idea is really difficult to implement. So my colleague is thinking about new ideas about how to implement ensemble method, because we previously we will think this should be quite straightforward, but then because four teams using different method, and then if we want to do the coding, it just become too complex and impractical. So we were think my colleague is just thinking about something that is practical for and let's see whether the ensemble method will perform better than any individual method. Yeah, collaborative efforts to improve the models. Yes, we have got lots of helps from the other teams, but we have got the SNP sets, and then we discuss about how to do the cross validation, etc. But then everything was done, but it didn't really uh, work yet.
Yeah.
We hope that it will be better in once we start combining the SNP sets, and yeah, maybe it will improve. Okay, next slide.
Oh, uh,
As I said, we will try to by combining and deleting existing pre-selected SNP sets. So I think the key problem here is still the SNP sets, and probably we will also do this sequential selection on existing SNP sets, so that we will have really the independent information. And then I hope that this will improve based on the current results. Uh, and also we are thinking about maybe stratifying to other covariates instead of the drugs may improve the performance, maybe, but just on the leaderboard. I'm not sure whether this will improve the performance on the corona data. So, but maybe the result can help us to understand why the predictions are better or not. Okay, so that's all our what we have done.
that's a
And
lot of work. No, thank you. And I appreciate you being so candid about describing it across all of the other groups. Does anyone have any questions or comments or suggestions for Lou? You can raise your hand or type a question in to the box, and I'll unmute you. And that will continue to be true through the entire webinar. Please just let us know if you have any comments. Okay. Well, thank you, Lou. I'm gonna mute you, and I'm gonna move on to Tao, who's representing. Who's representing Team Tracy. If I can figure out how to work my computer. Okay, Tao, I'm unmuting you now. Hello, I think you're there.
Hello? Oh, hello. Can you open the slides?
Yes.
Okay, so for the past 2 weeks, we didn't do much because I was away for a few days. And we gave multi-probe a try. It was finally updated on their webpage. So please go to the next slide. Yeah, so multi-probe is it's a method that has improved SNP-based prediction for complex traits. It has been shown to perform better than the blob method. And please go to the next slide. So blob stands for best linear unbiased prediction. It's widely used for predicting complex traits, and recently more so in human genetics. And the blob method has one severe limitation, that it can only implicitly assume that all SNPs have the same effect size. So the multi-blob method is better than the blob method because it can incorporate SNPs that correspond to distinct groups with different effect size variance. And recently, the author has updated the LDAK software to version 4.2, which formally implements the multi-blob method. So please go to the next slide. So I gave it a try. This is what I did. So I took the 20,000 immunology-related SNPs defined by the KGG pathways and put them into the multi-blob method. So this is a genetics-only model. So you don't expect it to outperform those models, which considers the clinical variables too. So for sub-challenge 1, the correlation is 0.13. For sub-challenge 2, the area under curve is slightly more than 0.5. So the conclusion is that multi-blob does not perform very well yet. However, because I just gave it the first try, so we don't know if I put in a better SNP set, it will better or not, and what will happen if I combine the multi-blob prediction with other predictions that also relies on the clinical data. So that's my future direction of research. I will give it a try in about 1 week and see what happens. And I have also attached the code in the next slide. Please go to the next slide. So this is the shell script that I use to run multi-blob, and I welcome anyone else in my group or in other groups to give it a try also. That's what I have so far.
Well, thank you for that, and thank you for providing your code. If it's all right with you, I'll circulate your slides so that people can use it if they're interested. Is that all right?
Yep.
Okay. So in terms of feature selection, you're using. If I go back, let's see if I can. Sure. You're just starting with the 20,000 KEGG-related or KEGG-identified immunology-related SNPs, but you're thinking that feature selection is going to play an important role. Would you be?
Yeah, I guess so. Maybe. Yeah, I think maybe I will put in all the SNPs, or maybe a few hundred or or a few dozen SNPs that are most related to the to response to the RA disease.
Uh-huh.
Yeah.
Uh-huh. As a subset of these 20,000 SNPs, or independent of that?
No, that's independent. Those found by literature mining.
Yeah. It'd be interesting to see what that does.
Yeah, and also there are some many parameters in the multi-block method. Maybe it's worth the try to tune those parameters and see what happens.
Mm-hmm. Mm-hmm. And then, yes?
It looks like there's a question from Victor Bellin. Victor has his hand up.
Okay, can you unmute Victor?
I can do that.
Okay.
Victor, are you there?
Hi, you hear me?
Yeah.
Yes, we have here a question. That where did you find the list of the SNPs of 20,000 SNPs?
Sorry?
How you select them?
How I said it. Okay, so I took down the KGG pathways and find the immune-related genes in those KGG pathways, and I find the leaves that are very close or just located within those immune-related genes, and these are the 20,000 immunology-related SNPs that I found. Yeah. Hello.
Great, thanks.
Yes. Okay. Thanks.
Thank you.
Okay, so then, and I wonder, Tao, if you could speak about your experience working in the group with the 3 other teams in your group. How has that benefited your team?
Yeah, I think it's quite inspiring to work with the other teams in this group. I learned a lot from their approach and their way of thinking. Although I haven't been able to incorporate specifically their methods or their SNP sets in my method yet, but I think it will help me a lot.
Great. And so I guess my last question is, how feasible do you think this question is? What's your opinion on the contribution of genetics to the prediction of this phenotype?
I think it will contribute, although not as much as the clinical variables. However, if you don't use the genetics information, you will definitely have a worse predictor. Yeah.
Yeah.
It definitely contributes, although not as much as you would expect.
Yeah, fair enough. Okay, thank you very much. Are there any other questions? If there are other questions, please, again, raise your hand or type your question in. So thank you very much, Tao. I really appreciate that. And I'm going to move on to Wanfeng. Let me see. I'm going to unmute you. All right. Hello?
Hi.
Hi.
So we prepared some versions for submission, but we didn't figure out that we need an invitation to submit until last week. So right now we're still waiting for the approval to submit to the leaderboard.
I apologize for that. That is my fault. I will.
But we actually did some cross-validation anyway. So what we did is we used other group SNPs in our model, and it looks like the overfitting is pretty severe based on our cross-validation. So honestly, I do not expect them to work very well in a final test set, but let's just see how they work in the leaderboard first. So yesterday I talked with Lu. So before that, we didn't realize that they were also using Gamma in their previous submission. So the first author of Gamma was recruited to University of Michigan this year. So I actually had a chance to talk with him several weeks ago. And we tried his software on our machine. So based on my discussion with him, it's unlikely to perform very well on this data set.
Huh.
So I just recently, actually last week, I implemented boosting, try to boost all the methods together. So I don't know if there's something wrong with my implementation. So far, the boosting result is always only slightly better than the worst worst performing one. So I'm still trying to figure out whether there's a bug or if it's just some aftermath of SVM. Since SVM is higher form SVM is just there's some similarity between boosting and SVM. So that's the progress on our side.
Very good. And so, what are you using for your SNP set? Are you incorporating the SNP sets from other members in your group, or are you focusing on the ones that you'd identified in the team phase?
So we use them one by one. That's why we will have 8 versions.
Ah.
Yeah.
Interesting. Okay.
Separately. Oh, actually, I don't know what will happen if we put them together.
Yeah.
It's too much. But can you suppress the contribution of SNPs?
Yes.
Yeah, well, we probably can try that as well. Yeah.
Yeah.
Yeah. Okay.
All right. Thank you. Are there any questions? If not, we'll move on to Javier. Let's see. I'm going to unmute Javier. Hello?
Yeah, hello.
Hello.
I have to apologize because the last 2 weeks we have not been able to advance as Daniel is out of the lab and I'm with other deadlines. But as a summary, as the strategy that we were following was, as in the team phase, we saw that the certification to select the SNPs was important, and we did it by drag. In this collaborative phase, we proposed to stratify also not only by the treatment, the drug, but also if the patient takes methotrexate or not, and which is the baseline DAS of the patient. So patients with low baseline DAS, medium baseline DAS, and high baseline DAS. This is what we exposed in the last seminar, and these association measures were uploaded to the Dropbox of the group. And I just did a few tests of submissions just using this clusterization, using this with patients treated with methotrexate and with each specific drug using a different set of SNPs. And yeah, and the other part that we were doing in the collaborative phase was to select the SNPs based on protein-protein interaction networks and metabolic networks. And I have not worked now in the collaborative phase on it. Daniel has done it. And he has used the Guild software to score the proteins in the network associated with arthritis. I also know he used Reactome, a metabolic network. I know the numbers of SNPs that he has used. So from all the SNPs, he has a final list of 9,000 SNPs, but we still don't have results on how these SNPs perform on the leaderboard. So I have to apologize because we have not been able to advance more.
No, that's August is a difficult time for any sort of analysis, so that's understandable. So tell me, what has your experience been with the collaborative approach and interacting with the other groups? How has that influenced your thoughts in your modeling?
Well, I think it's useful, but as I say, I'm not really involved in the collaborative effort. It's Daniel, but he's not now in the lab.
Gotcha.
But I think the collaboration and sharing of SNPs and methods is good.
All right. All right, thank you very much, and thanks to all in your group. Let's move on to the group that is, I think you're unnamed, the group led by Matthias, Daniel, and Eric. So I think we'll go in that order. Matthias, I'm going to start with you. I'm going to unmute you and ask you to talk to us a little bit about what your experience has been in this community phase. So, okay, there you are.
Yeah, I think the group that is unnamed is actually quite a cool name. So yeah, from my side, I can report, and I'm sorry I don't have any slides to present. But like most others, I have to say, unfortunately, that whatever genetic model I tried, it did not perform significantly better than a clinical data-only model. And so far, in the last weeks, I focused mainly on working with eQTLs. I derived eQTLs for different lists of genes, both from prior knowledge like FarmGKB, but also from other based on other rationales. But like I said, just feeding the raw SNP data to the classifier I use, I'm mostly using gradient boosting ensemble classifiers. It didn't really improve upon a clinical-only model. So I think the last thing that I will try to do in the remaining time left for the challenge is to work on calculating some kind of compound score for each gene, so that there's not raw data fed into the classifier for all the SNPs, but a compound score for the eQTLs for each gene. Unfortunately, I'm still rather undecided what the best approach for doing this would be. But yeah, I'll try and see if this improves in any kind. Yeah, that's it for me.
Very good. And if there's anyone who has any suggestions for Matthias, please drop him a line. So tell me a little bit about your group. How have you guys been collaborating, and how has that influenced your decisions on how to move forward?
So I think, like the other group, we collaborated in a rather loose manner. So we shared ideas and also lists of genes and SNPs. We didn't really share code or work together on coding as much. Mm-hmm. But I think it is still very valuable, even though the collaboration was not very intense in that respect. Sharing of ideas and basic approaches was still very valuable, and I think it really helped me to understand the problem domain of pharmacogenomics a lot better than I did before, even though we can't really show much of a success in this specific case. I think I have a much better understanding now of the problem domain in general than I did before.
Oh, good. I'm glad to hear that. All right. Thank you very much. We're going to move now to Daniel. So let's... Okay, Daniel, I'm unmuting you. Hello.
Okay, so we haven't been able to make too much progress over the last 2 weeks.
Daniel, we're having a hard time hearing you. Can you get a little closer to the microphone?
Yes, sorry about that.
Oh, that's great.
We haven't made too much progress over the past 2 weeks, or we haven't made a new submission. We're still working on some code, but I can answer the 4 questions that you sent us.
Great.
So the use of biological priors for feature selection, we didn't use biological priors. We actually just used prior knowledge from FarmGKB to select some SNPs. Though we're planning to use some eQTLs in the future. Now, for the polygenic modeling approach, we're working on a neural network, but we still haven't finished it yet. So once we get more results, once we start the tests, we can provide some results. For the use of ensemble methods, we haven't considered it yet. We're actually still interested in working on the single models first before we consider ensemble methods. And for the collaborative efforts, I just like Matthias has said, I found it pretty valuable. We did share a lot of ideas, and even recently, Matthias was able to help us with getting eQTLs, the genes that were associated with SNPs in our FarmGKB list. So it's very useful to have team members who have a lot more experience with retrieving different sorts of biological information, because we don't have that skill.
Good. So tell me, how has your group considered how they might combine efforts at the end? You know, you're all still working on your individual approaches. Has there been any consideration about how those may intertwine at the end of this phase?
So we haven't considered that yet, and one of the reasons for that is we haven't been able to come up with a method that has been significantly improved with SNPs, genetic information. So until that point, it doesn't make sense to spend too much time discussing how to integrate the multiple predictions when we haven't seen any good predictors yet.
Yeah, fair enough. Okay. All right. Thank you very much, Daniel. Move on to Eric. All right, Eric, I'm unmuting you. Hello.
Hey. So sadly to report, I also haven't been able to make much progress. I guess what I've been focused on is kind of making compound features from the SNP data, because including the raw SNP data hasn't been very successful. Yeah, so some of the features I have, so we predicted looking for SNPs that were damaging mutations, and then we aggregated those damaging mutations to pathways. And so for each of the patients, we have how many damaging mutations they have in each of the reactome pathways. So we tried including that as features, but that didn't really improve the models either. So if any groups want that data, I can send it to you. Similarly, we still have the multidimensional scaling components, if anybody wants to try to use that. So that's just roughly like a dimensionality reduction technique, but similar to a PCA, except it's better for binary data. Yeah, and so that is roughly correlated with the response to treatment, but it doesn't improve the model. And then another thing that we took the Wellcome Trust Case Control Consortium, so they have a rheumatoid arthritis cohort. And so we built a model to predict overall risk for rheumatoid arthritis, and then predicted on the data that we have, and we found that the patients that have a higher risk for rheumatoid arthritis do respond better to treatment. But again, including that into our model doesn't improve it any significantly more than just a clinical-only model. So that's been kind of our finding so far. But if any teams want those features, just send me an email and I can send it to you. Maybe you will have better luck modeling than I did. Yeah, and then yeah. So, anyways, to go over the questions, I guess, yeah, the use of biological priors for feature selection. I think obviously it's a great idea, but here we're mostly doing it just by pulling down from known SNPs from either GWAS catalog or EQTL databases. Again, polygenic modeling approaches, I think was a great idea. I think that's what we're kind of getting at with HLA typing and the MDS analysis. So kind of combining multiple SNPs into a single feature, I think is a good idea. Yeah, for the ensemble methods, again, I think it's a good idea, but we're using a tree-based approach, which is an ensemble method, and yeah, so in our modeling that works the best, and so we've continued to use that. So it is an ensemble method that we're using, and yeah, collaborating's been great. I've loved hearing ideas from our other group members because they have a lot of really good and clever ideas. So yeah, that's been great. So.
All right, thank you very much, Eric. Much appreciated. So we'll finish up with, yeah?
Can I ask a question before we move on?
Oh, please.
I was curious about your use of the Wellcome Trust data and your risk looking at the risk score in these cohorts. Did that give you any information above and beyond baseline DAS, or is that basically picking up the same? Are people with the higher risk scores basically just the most severe cases?
Yeah, they're just the most severe cases, so it's not picking up any new information, but it is picking up basically baseline DAS. So yeah, the patients that have a higher risk for rheumatoid arthritis have more severe disease. So yeah, but the way we did it, we just pulled down the Wellcome Trust data, and then we imputed their genotypes to the loci that we had in our data, and then we built just a random forest model based off those loci.
Great, thank you.
Yeah.
Okay. Oh, there's a couple of questions. There's some hands raised. So We'll start with Daniel. I'm unmuting you, Daniel. Oh, you're muted on your side as well. Dan, there you go.
So it's actually not Daniel, it's Anna Goldenberg. We are one team. I have a couple of questions, just kind of thought process seeing this challenge unfold in terms of collaboration. We do exchange a lot of information with the other teams that are part of our larger team, but I wonder whether there could, like, I don't see why we are not exchanging all the information together, like all the teams, because the only information that we are hearing from them are on this 2-week basis, and I think hearing about something that they have tried, for example, they have tried the FarmGKB genes that we are planning to try now, it would be useful to know about this information. So I think, I don't know, at least for the future challenges, I would like to see that all the teams, there is a forum of open discussion and everybody is encouraged to share their findings as soon as they find them. I think it would be beneficial. And another question that I have, well, I guess the first one was not a real question, but a question that I have is, so far in this challenge, it looks like we really are not finding any strong effect beyond really good models for clinical data. So my question is, what will be the result of the challenge if that is what will be the ultimate finding?
Are you asking in terms of analytical conclusions or
publications, conclusions, publications, like the outcomes will be seen by the world, I guess.
Yes. So I think that there will certainly be a publication, no matter whether we identify genetic signal or not, because that is, of course, itself a conclusion. Having some of these benchmarks, looking at what we can say on top of clinical models will help us to do that. And then I think the social experiment that's going on as well with both the traditional challenge phase and this community phase will be integrated into that publication. And so writing that publication and engaging all of you in that process is, I guess, the next phase of the challenge. Something it's time to think about with a little more detail.
I'm really happy to hear that there will be publication either way, not because of the worth of a publication, but because I think it's really great to see if we really tried hard and didn't find genetic signal, that maybe it's not there. And I think there are not enough such publications saying, well, maybe you cannot really predict what genetic signal, and people should stop doing some of these studies and believe that the genetic signal is cannot always be found. So I think having a high-profile publication, having this result will really be beneficial to the science.
Good. And with respect to your first comment, I'm pleased to hear your feedback on that. You know, we're looking for ways to engage the groups, and this is the first time we've had a community phase, and so certainly it's not running perfectly, and there's always room for refinement and improvement. And so it's really useful to hear your feedback and you know, that of anyone who cares to give it to help make sure that it runs you know, more efficiently the next time we try this.
Thank you.
Thanks. There was another hand, I thought. Looks like it's down. Okay. So let's then move on to Victor. Victor and his team, Team Lucia's, have been focused on building a model entirely on clinical covariates that we can use as a benchmark to really assess whether these other approaches are finding any signal at all. And so it is in the interest of a publication, as we just described, we need to be able to say definitively whether there is or there is not signal here. And so Victor's going to describe to us what he's been doing, and really I encourage all of you have been running your own clinical covariate only models. So please provide Victor with feedback on what he's doing, because he's representing all of us here. All right, Victor, I am unmuting you with, you're muted on your side as well. So you will have to unmute yourself too. There you go.
Okay. So we have not been really active until 1 week ago that I came back from my holidays. So we're still in process. We have just starting submitting the first models. But our first approach is to compare different ways to use the random therapy variables. So we are looking at what gives us best results if you use them as a variable, not use them at all, or use them to stratify the data. And that's what I'm looking right now, just using the same model that we used in the previous phase. So support vector regression, that's not much. But also we're doing some bit optimization of the parameters of the models and the way we combine the kernels. But the point of these first steps is just to decide how we can better use the drug and cotherapy variables. So this is not already done, but we're working on that. And so next steps that we want to look further is going to be to look to ensemble models, so random forest, boosting, and look a way to, and if it is possible, look a way to integrate other methods, not just random forest, so more complex methods, more prediction from support vector, Gaussian process, whatever. And well, once we get those first results of the comparisons, we will share it. We will share it through the mailing list that is available, so you will receive it. I hope that it will be finished by the end of this week. I will have to process so that I can share with everybody. That's all.
Excellent. So let's see. If we look at the leaderboard, we can see that you are starting to submit, and so I think it'll be really useful for when you're satisfied with your results for you to pass them around to all of the other individuals, because as I said, everybody's been using their own specific approach and looking at the effects of covariates in it, and so I think there'll be a lot of useful feedback. I think our collective goal is to make sure that we have the best possible clinical model that we can use as a benchmark to assess the genetic contribution of these other approaches. So we look forward to seeing what you have to circulate later this week, if possible.
Laura, it looks like there's a question from Daniel. And it looks like, or Dana or Anna, are you guys muted on your side? You're not muted on our side.
So I was curious to know what was the best clinical leaderboard score that you received so far?
So in the leaderboard there is only 2 submissions actually. So the one you're seeing in position 31 is just using one model with the following variables, which is baseline, sex, age, and cotherapy. And well, for each variable we have calculated a different kernel and combined them. So for the one is that, I mean, we have actually only one other submission for the moment, that it's just using baseline, sex, and age. So it's most basic model. But so for the moment, that is here in position 57. You can see I submitted 2 times by mistake. But yes, for the moment it's just that. But later I have to submit. I should have some more results from my experiments, so I will submit them. And well, the name of the if you want to look before I share the information, if you look at the name of the submission, it will be more or less the same. So MTX bar says that MTX is being used as a variable. If I put a set of separate, it's being used to stratify, and also the code name will be track. So you can know whether I'm using drag or not, and if I'm using it to separate or to a variable, if you are curious before. But
okay, and I have one more question.
I don't know if it's clear.
Have you found age in your predictions?
Sorry, I didn't hear you well.
Have you found using age as a variable useful in your predictions?
So for the moment, we have not tested, but that's a thing to test it, I think.
Okay, thank you.
Yes. So I will test it. I think it's not too much work. So thanks for the idea.
Okay, thank you, Victor. So I imagine it'll be interesting to see what the final correlation is for the clinical only model, and we can use that to assess these other models. So, okay, so we've heard from all the teams. Thank you all for taking the time and energy to tell us how you're doing. I'd like to end by asking Eli Stahl, who knows this data better than any of us, to give his opinions on what he's heard and reflect on where we are. Eli, are you there?
Yeah. Can you hear me?
Yeah.
I think that it's really impressive work that people are doing, and I really appreciate it. I just hope that I can just say a couple of words about the feature selection and the polygenic methods. So for feature selection, it really seems like the different teams or groups, or I'm not sure what they're calling, but what we're calling them, but at the highest level, the different groups are specializing in some of the biology for feature selection that I at least intuitively thought would be interesting. And so I guess I second the comment that was made that interaction between the groups at the highest level could be worthwhile. And specifically, I'm talking about when we had the second slide set which actually used the multi-blip method. They had this immune-related gene set and that they had mapped SNP2 and were using. And then the third presenter discussed progress in protein-protein interaction networks. And then I believe it was Matthias who mentioned that he has been working with EGCL data. So those 3 areas right there, one might think that we should put together at some point. Again, it's really great to see people are acting on these feature selection approaches, although I guess we're not seeing any game changers, so that's perhaps disappointing. But to just go a little bit further in the polygenic method, and I wish that we could all sort of talk at the same time in a more sort of in an easier way, but my understanding is that the second slide set with the multi-BLUP method is truly a polygenic method, but at least the first approach, describing using the Gemma software and the third method also using that software, I didn't understand that those were polygenic. I thought perhaps they were not, and specifically for the first slide set, right, there was a set of 29 features that were being tested, and the original published use of Gemma was a mixed model GWAS, where you would do this polygenic analysis, but use the phenotypic residuals from a polygenic score that would be similar to BLUP predictors, but use the residuals from that and do GWAS on that. And then perhaps a straightforward use then of the software would be to do that and then take the top SNPs, for example, 29 SNPs, and then put those into a predictive model. Is that more like what has been done by both groups 1 and 3? So if so, it's not polygenic at all, right? The multi-BLUP method is definitely, and the starting point of 20,000 SNPs, I think, is actually not a lot for a BLUP predictor to be developed. One could start with more, and one could start, for example, by adding features that map to those immune genes, but then adding to those with significant EQTL effects, or in protein-protein interaction network with any number of seed gene sets that one could imagine, and then just to kind of ask, maybe that person who presented could potentially get back on for a second, but the 0.13 correlation that was reported, and that was genetics only, it was said. So is that actually potentially quite substantial then, particularly if the model was trained on delta DAS, not baseline DAS? So I am interested anyway to find out whether that does translate into an improvement in the genetics over and above clinical when they're incorporated into the same model. The one more thing I wanted to ask about is just the approach of adding the BLUP predictor as a feature into a model where you have say 29 or some relatively small number of features, adding those together in the ensemble predictor. That would be interesting to see if we have an opportunity to look at that. That's what I wanted to say. Thanks.
All right. Tao, do you, those last comments were directed at you and your efforts, so I'm going to unmute you and give you a chance to comment. There you are. Are you there? Tao? No.
Hello, can you hear me?
Yes.
Yeah. So so I agree that 20K SNPs is not a very big set. I will try to put in all the SNPs as my second trial, and also use like a gene interaction network or protein-protein interaction network to put in all the SNPs. As a first try, I tried 20K SNPs. It's the only reason is that it's a small set, so the multiple of software will run fast. Okay, so that's what I will do. And as for the correlation point, 0.13 is not very impressive. I remember from the first phase of this challenge, people who submit genetics only model can get as high as 0.3. I mean the correlation for the first challenge. So it's not substantial yet, but I think there is definitely room for improvement. Yeah. So I will try out, like tune the parameters, put in difference SNP says and see what happens. We will see. Yeah.
Okay, thank you.
Yeah. So yeah, that's what I have.
All right, Eli, any other comments?
I mean, not at this time. I hope that we get the chance to continue to have at least as much back and forth, though. I hope that actually that we end up using the Google group more. And I did want to suggest maybe putting the slides actually on the Google group or otherwise making them available to all of us would really give us more to go on in terms of commenting towards each other and stuff.
Yeah, and I'll look into the dates and see if there is time to have one more of these interactions. And if so, I'll look for a different venue than GoToWebinar. I actually think there might be an alternative that allows us to all speak with each other a little bit more freely, which would be, I think, more useful at this point. Right, so also I'll make sure to circulate the slides, and I am recording this, so I'll circulate the recording as well, and some instructions for how we're going to wrap this up over the next 4 weeks or so. All right, well, thanks everybody again for your attendance and for sharing your ideas and thoughts with us, and yeah, happy modeling. Thanks.